Hello there! Welcome to Merivit Education Center, and this is Science with Master Rick. Today we're going to be learning about cell division, most specifically on mitosis. Cell division is a process by which a parent cell divides into two or more daughter cells. Cell division usually occurs as part of a larger cell cycle. There are several types of cell division depending upon what type of organism is dividing. For unicellular organisms, cell division is the means of reproduction. Whereas in multicellular organisms, it is the means of growth and maintenance of tissues and organs. These are the two types of cell division. Number one, they are mitosis, and number two, meiosis. What is the difference between mitosis and meiosis? Mitosis is a process that occurs during the cell cycle. The role of mitosis in the cell cycle is to replicate the genetic material in an existing cell, known as the parent cell, and distribute that genetic material into two new cells known as the daughter cells. In order to pass the genetic material to the two new daughter cells, a parent cell must undergo cell division, or in this case, mitosis. Mitosis results into two new nuclei which contain the DNA that eventually become two identical cells during cytokinesis. The main purpose of mitosis is to accomplish cell regeneration, cell replacement, and growth of living organisms. Mitosis is important because it ensures that all new cells that are generated in a given organism will have the same number of chromosomes and genetic information. Mitosis occurs in 5 discrete, consistently consecutive phases. Number 1, interphase. Number 2, Prophase. Number 3, Metaphase. Number 4, Anaphase. And number 5, Telophase and Cytokinesis. During the 4 phases of mitosis, nuclear division occurs in order for one cell to split into two. Sounds simple enough, right? But different things occur in each step of mitosis, and each step is crucial to cell division occurring properly. That means successful cell division depends on the precision and regulation of each phase of mitosis. That is why it's important to be able to understand and articulate the role of each phase in mitosis overall. We can think of interphase as a transitional phase. Interphase is when the parent cell prepares itself for mitosis. This phase isn't considered part of mitosis, but understanding what happens during interphase can help the steps of mitosis make a little more sense. Interphase occurs prior to the beginning of mitosis and encompasses what's called stage G1, or first gap, stage S, or synthesis, and stage G2, or second gap. Stages G1, S, and G2 must always occur in this order. The cell cycle begins with stage G1, which is a part of interphase. So how does the parent cell prep itself for mitosis during interphase? During interphase, the cell is busy growing. It's producing proteins and cytoplasmic organelles during the G1 phase, duplicating its chromosomes during the S phase, then continuing to grow in preparation for mitosis in the G2 phase. In the cell cycle, interphase doesn't just occur before mitosis, it also alternates with mitosis. Now it's important to remember that this is a recurring cycle. When mitosis ends, interphase starts up again. In fact, in the grand scheme of cell cycle, mitosis is a much shorter phase than interphase. Prophase is the first step of mitosis. Now this is when the genetic fibers within the cell's nucleus, known as chromatin, begin to condense and become tightly compacted together. During interphase, the parent cell's chromosomes are replicated, but they aren't yet visible. They're just floating around to form the form of loosely connected chromatin. During prophase, that loose chromatin condenses and forms into visible individual chromosomes. Since each of the parent cell's chromosomes were replicated during interphase, there are 2 copies of each chromosomes now in the cell during prophase. Once the chromatin has condensed into individual chromosomes, the genetically identical chromosomes come together to form an X, or an X shape called sister chromatids. The sister chromatids carry identical DNA and are joined together at the center, at the point called the centromere. The centromeres will serve as anchors that will be used to pull the sister chromatids apart during the later phase of mitosis. And that's what's happening inside the nucleus during prophase. After the sister chromatids form 2 structures called centrosomes, they move away from each other outside of the nucleus. As they move to opposite sides of the cell, the centrosomes form something called the mitotic spindle. The mitotic spindle will eventually be responsible for separating the identical sister chromatids into 2 new cells and made up of long protein strands called microtubules. Metaphase is that phase of mitosis that follows prophase and prometaphase and precedes anaphase. Metaphase begins once all the kinetochore microtubules get attached to the sister chromatids centromeres during prometaphase. Prometaphase is often referred to as late prophase. Prometaphase is the phase of mitosis following prophase and preceding metaphase. The short version of what happens during prometaphase is that the nuclear membrane breaks down. So here's how it happens. The force generated during prometaphase causes the microtubules to start pulling back and forth on the sister chromatids. Since the microtubules are anchored at opposite ends of the cell, their back and forth pulling on different sides of the sister chromatids gradually shifts the sister chromatids to the middle of the cell. This equal and opposite tension causes sister chromatids to align along an imaginary but very important line trailing down the middle of the cell. This imaginary line dividing the cell is down the middle is called the metaphase plate or the equatorial plane. Now, in order for metaphase to progress on to anaphase, the sister chromatids must be equitably distributed across that metaphase plate. If they are, the cell gets the green light to move on to the next phase of mitosis. During anaphase, the centromeres at the center of the sister chromatids are severed. Remember how the sister chromatids are attached to the mitotic spindle? The spindle is made up of microtubules which start shrinking during this phase of mitosis. They gradually pull the severed sister chromatids towards opposite poles of the cell. Anaphase ensures that each chromosome receives identical copies of the parent cell's DNA. The sister chromatids split apart in the middle at their centromere and become individual identical chromosomes. Once the sister chromatids split during anaphase, they're called sister chromosomes. They're actually more like identical twins. Now, these chromosomes will function independently in new, separate cells once mitosis is complete, but they still share the same and identical genetic information. Now, finally, during the second half of anaphase, the cell begins to elongate as polar microtubules push against each other. It goes from looking like one round cell to, well, more like an egg, as the new chromosome sets pull further away from each other. At the end of the anaphase stage, chromosomes reach their maximum condensation level. This helps the newly separated chromosomes stay separated and prepares the nucleus to reform, which occurs in the final phase of mitosis, the telophase. Telophase is the last phase of mitosis. Telophase is when the newly separated daughter chromosomes get their own individual nuclear membrane and identical sets of chromosomes. Once the daughter chromosomes have fully separated to opposite poles of the cell, the membrane vesicles of the parent cell's old, broken down nuclear envelope form into a new nuclear envelope. The new nuclear envelope forms around the two sets of separated daughter chromosomes, creating two separate nuclei inside the same cell. Now, you might think of the events of telophase as a reversal of the events that occurred during prophase and prometaphase. Remember how prophase and prometaphase are all about the nucleus of the parent cell starting to break down and separate? Now, telophase is about the reformation of the nuclear envelope around the new nuclei to separate them from each cell's cytoplasm. Now that the two sets of daughter chromosomes are encased in a new nuclear envelope, they begin to spread out again. When this occurs, it is the end of telophase and mitosis is complete. Cytokinesis is the actual division of the cell membrane into two discrete cells. At the end of mitosis, there are two new nuclei contained within the existing parent cell, which has stretched out into an oblong shape. So at this point, there's actually two complete nuclei hanging out in one cell. So how does one cell become two cells? Cytokinesis is responsible for completing the process of cell division by taking those new nuclei, separating the old cell in half, and ensuring that each of the new daughter cells contains one of the new nuclei. During cytokinesis, a contractile ring made of protein filaments develops where that metaphase plate used to be. Once the contractile ring forms down the middle of the cell, it starts shrinking, which pulls the cell's outer plasma membrane inward. You can think of it like a belt that just keeps tightening around the middle of your cell, squeezing it into two sections. Eventually, the contractile ring shrinks so much that the plasma membrane pinches off and the separated nuclei are able to form into their own cells. The end of cytokinesis signifies the end of the M phase of the cell cycle, of which mitosis is also a part. At the end of cytokinesis, the division part of the cell cycle has officially ended. And that ends our exciting topic for part 1 of cell division in mitosis. And as always, remember, science is cool.